Thank you, Professor Lin Lu and the organizing committee for the kind invitation to give a presentation today about the plant homeodomain finger protein 21B as a novel modulator of social memory. I have no conflicts of interest to disclose. I'd like to acknowledge people from SUNY Upstate in my lab and also collaborators who have helped in this project. People from Australia, from Summary, also made important contributions to this work. We have had many collaborators in the past, and some of them continue to collaborate with us. Plant homeodomain motifs, they govern plant development, especially the flowering of plants, and they are epigenetic readers. Epigenetics are mechanisms for regulating gene activity independent of DNA sequences. There are three types of epigenetic molecules: writers, erasers, and readers. And the PHF21B is an epigenetic reader. It means that it binds to histone modifications and regulates gene activity and protein production. In this cartoon, you can see here the histones and then some modifications. The erasers, they remove histone modifications. The writers, they add histone modifications. And the readers bind to the histone modifications and decode the meaning of those modifications. They can activate genes or repress genes and change protein production. Plant homeodomain finger proteins is a diverse set of epigenetic readers. They contain a PhD domain, which is about 50 to 80 amino acid residue containing a zinc-binding protein. This regulation of PhD fingers proteins is implicated in cancer, immune, cardiac, and neurological disorders. Today I'm going to talk about the PHF21B gene, which is located in chromosome 22q13, and deletions of this chromosomal region gives rise to the Phelan-McDermid syndrome, which is characterized by intellectual disability, hypotonia, absent or delayed speech, and autistic-like behavior. Little is known about the function of this gene. In 2016, our group published that rare variants in this gene were associated with major depression. Later, a group in Germany published that PHF21B controls an epigenetic program for neurostem cell differentiation during cortical development. But the functional role in the adult brain is unknown. Our work was a relatively small study in Mexican American that associated the variants in the PHF21B with the major depression risk. And we also found out that this gene modulated the stress response. Here's the flow diagram for the genetic study. We used an exome chip and we studied common variants and rare variants in Mexican Americans and replicated in the European ancestry cohort. We also performed the whole genome sequencing analysis of 25 samples, 10 Australian samples, and 15 Mexican American samples. And here is the Manhattan plot for the Mexican American cohort. It's roughly 200 patients and 200 controls. A rare variant analysis in the European cohort showed the replication of the PHF21B in the risk for major depression. We also performed quantitative gene expression analysis for variants in the PHF21B gene using the Brainiac web server from the UK Expression Consortium. We were interested in understanding the role of PHF21B gene in the brain, so we generated a transgenic animal to study that. Aditya, a student in our lab, performed Western blots to characterize the expression of PHF21B in central nervous system, tissues, and peripheral organs, and found that PHF21B is highly expressed in the central nervous system. In rats submitted to chronic restraint stress, we found that animals that were resilient to chronic restraint stress had decreased the PHF21B gene expression in the hippocampus. Basu et al. Also had previously reported that PHF21B imprints the spatiotemporal epigenetic switch essential for neural stem cell differentiation. We generated the mouse model for PHF21B knockdown in Australia and brought it to Zuni Upstate to study its behavior and brain function. Using CRISPR, we deleted the exon 4 of this gene to generate a knockdown mouse model. The exon 5 encodes for the PhD finger domain. The deletion of exon 4 was confirmed by sequencing, and the off-target analysis did not give any off-target site. The knockdown of PHF21B was confirmed at messenger RNA level and protein level by Western blot and immunohistochemistry. Here in this image, you can see PHF21B in green, and here's the knockdown showing much decreased PHF21B expression. Our postdoc in Eunice Jean spearheaded most of this work. She performed behavioral testing. The PHF21B knockdown animals had a normal locomotor activity in the open field, elevated plasmase, and Rota road. They had increased self-grooming in the open field. These animals did not exhibit any anxiety-like behavior. In the open field, the time in the center was not different from the wild type, elevated plasmase, and the time in the open arms and open arm entries was not different. And they also did not exhibit any behavior despair in the tail suspension test, for swimming test, or anhedonia in the sucrose preference test. The PHF21B knockdown animals also had normal memory and learning in the Morris water maze. Interestingly, these animals exhibit increased preference for social novelty in the three-chamber test. So they had increased social novelty index. We complemented the 3-chamber social test with the 5-trial social memory test and find out that these animals do not habituate to the familial mouse. And so that showed a problem in social memory. The PHF21B knockdown mouse had the thinner cortex. Here is the cortex of wild type and the knockdown and the quantification. So they had a decreased thickness in the cortex. They also had the reduced neurogenesis, here shown by double courting positive cells. The knockdown had the reduced number of double courting cells, but then neurons, the number of neuron cells was not significantly different from the wild type. They had decreased number of astrocytes here. The knockdown animals also had decreased hippocampal synaptic protein expression, the PSD95 and GRAR1 were decreased in the synapses. And the glutamatergic neurotransmission was also decreased, here shown in the blue dots. We performed transcriptome analysis in the hippocampus and found that many genes were downregulated in the knockdown mouse. A lot of these genes were involved in neurotransmission, and we validated some of our findings, the RNA-seq findings, using RT-PCR. These analyses were done by the Molecular Analysis Core facility at SUNY Upstate. As I discussed in the beginning of the talk, PHF21B is an epigenetic reader, and so it regulates transcription by interacting with the histone modifications. And we found that it interacts with H3K9 acetylation, H3K9 MA2, and CRAB. The levels of H3K9 acetylation, H3K9 MA2 were increased in the knockdown mouse. Phosphorylated CRAB was decreased, the total CRAB was unchanged, and the ratio phosphorylated CRAB by total CRAB was decreased in the knockdown animals. PHF21B knockdown mice had decreased the hippocampal GLUN2B and GRIN2B levels, and GLIA1 was not significantly different. We purified a recombinant PHF21B protein and used to probe an array with several histone modifications, and found that it binds with a higher specificity factor to H3K36MA3. And this is a novel finding that was not described before. And the knockdown has decreased the expression of H3K36MA3. So we could conclude that PHF21B regulates transcription by its interactions with H3K9 acetylation, H3K9 MA2, CRAB, and also it interacts with the H3K36MA3. the interaction with the H3K36MA3 still needs to be detailed, and we still need to understand if it really promotes activation or repression by binding to H3K36MA3. So here are some take-home messages. PHF21B is an epigenetic reader. It functions as key upstream regulator of gene expression events linked to neurotransmission regulating social memory. There is a reduced expression of synaptic proteins and impaired long-term potentiation in the knockdown hippocampus. There's altered glutamatergic neurotransmission in the knockdown mouse. Transcriptome profiling revealed the differential expression of genes involved in synaptic plasticity processes. We characterized a potentially novel interaction of PHF21B with histone H3 trimethylated lysine 36. Histone modification associated with transcriptional activation and the transcriptional factor CRAP. Upcoming work will focus on CA2 and the frontal cortex, as these regions have specific roles in social memory. And PHF21B knockdown mouse have decreased GLUN2 clustering in CA2 and morphological defects in the cortex. In addition, future studies are needed to establish PHF21B as a candidate therapeutic target for underlying synaptic dysfunction that's widely prevalent in neurobehavioral disorders. So this is a cartoon which summarizes our findings, and these animals mainly have a problem in social memory, distinguishing familiar from stranger mouse. Thank you so much. That's the conclusion of my talk. Unfortunately, cannot answer any questions, but if you have questions you would like to email me, please do so, and I will respond to your emails. Thank you. Bye.